Hello everyone, my name is Prithvika K, student of Sri Venkateshwara College of Engineering, Bengaluru. I'm currently pursuing my engineering degree under CSE AI stream USN 1VE23CA036, and today's topic is about DNA and DNA fingerprinting course under the course of biology for CSE stream, course code BBOC 407. This topic we are going to learn about what is DNA and how it is presented, all about, and this presentation is all about DNA and DNA fingerprinting. Let's see now, where is DNA found. DNA is in every cell of every living thing. It is found with the chromosomes of cell. Chromosomes work to build proteins and acid in duplication or divisions of the cell. Now, seeing to the structure of DNA, where it has a unique structure and it allows it to be the hereditary molecule and allows it to store instruction for directing cells and its activities. DNA stands for deoxyribonucleic acid. It is a type of nucleic acid where deoxyribo means sugar molecule and nucleic acid is a macro molecule. This DNA structure has a double helix shape, which means the components present in DNA has bonded together, forming a twisted ladder shape. These four are the nitrogen bases which form DNA: adenine, thymine, cytosine, and guanine. Now let's see what is nucleotide. It is the backbone of DNA and it is formed by altering sugar and phosphides which held together, forming a strong bond. These ladder like structure is formed by those four nitrogen bases which held together by weak hydrogen bonds, namely adenine, thymine, cytosine, and guanine, where cytosine and guanine gets the hydrogen weaker bonds and adenine and thymine gets the hydrogen weaker bonds. How DNA look like. Yeah, like previously discussed, we saw that adenine always uh, gets the bonding with thymine, and cytosine gets its bonding with guanine. It can never be exchanged or be cross bonded. It always gets bonded with thymine, like adenine gets bonded with thymine, and cytosine gets bonded with guanine. How does DNA work? The four letters of DNA make up codons. These chemicals are repeated in various orders over and over. These codons make up genes. These genes tell cells how to make a protein that controls everything in a cell. Now let's see about DNA fingerprinting. Fingerprinting is a scientific method used to identify individuals based on their unique genetic makeup. It involves analyzing specific regions of DNA that vary greatly between people, known as variable number tandem repeats, short tandem repeats, and this technique was first developed by ALEC Jeffers in 1984 and has since become a crucial tool in forensic science, parentally testing, and genetic research. In forensic investigations, DNA samples from crime scenes can be compared with those of suspects to establish guilt or inconvenience. This process starts by extracting DNA from cells, followed by cutting it with the restriction enzymes, and even separated the fragment using these fragments create a pattern that is unique to each other, which makes a fingerprint. DNA fingerprinting is also used in identifying remains, solving historical mysteries, and verifying family relationships. It has applications in wildlife conservation to track genetic diversity among endangered species. This accuracy of DNA fingerprinting is extremely high, making it reliable method of identification. However, it must be performed carefully to avoid contamination or misinterpretation of results. Now let's see the conclusion. In conclusion, DNA deoxyribonucleic acid uh, is a fundamental molecule that carries the genetic instructions for the development, functioning, growth, and reproduction of all living organisms. Its structure is a double helix made up of two long strands of nucleotides twisted around each other, with the base pairs adenine thymine and cytosine guanine connecting them. This structure allows DNA to replicate accurately. During cell division, DNA plays a crucial role in heredity as it is passed from parents to offspring. Beyond genetics, DNA is used in various scientific and medical fields. In forensic science, it helps identify individuals and solve crimes through DNA. In medicine, it assists in diagnosing genetic disorders, developing gene therapies, and personalizing treatments. DNA is also used in agriculture to create genetically modified crops with desirable threats and contributes to evolutionary studies and helps trace human ancestry and migration patterns. Overall, this understanding and application of DNA have transformed science, medicine, and many aspects of modern life.